Welcome, thanks for coming. I'm Michael Shermer, the director of the Society, the publisher of this, the latest issue of Skeptic on terrorism. Is it an existential threat or just what it is? A way to get the U.S. government to spend $3 trillion. So we discuss the politics and the economics and the realities of terrorism in this, our latest issue, the quarterly publication of the Society, which you can get today by filling this out and joining for $25 instead of the usual $30, and you can get our latest issue here today. Now, as most of you know that have come here, we have a regular once or twice a month on Sunday afternoons. After today, our last event, regular event on Sunday afternoons is Leonard Mlodinow and his new book, which just came out, which just comes out this coming week, The Upright Thinkers: the Human Journey from Living in Trees to Understanding the Cosmos. And it's thinner than my book, so that's covering something like 1 million years of evolution there. That's pretty good in just a regular-sized book. Leonard's a very economical writer. He's quite good. Economy of words, I mean, not an economics writer. Economy of words. Anyway, so and then our last event of the first half of the year is our big conference at Caltech here in Beckman Auditorium across the lawn about the future of science and humanity in the year 2525. I thought maybe I should get the permission to play that song or something, but people of my previous younger than me probably don't remember that song. But anyway, so we have a Friday night party at the Westin. So all the speakers will be there: Lawrence Krauss, Richard Dawkins, and so on. And then talks all day on Saturday with the likes of Lawrence Krauss and Jared Diamond and John McWhorter, the linguist, who will be talking about what language we might be speaking in a century or 2 or 5 from now. Will it be Chinese? Will it be English? Will it be whatever? Something is different from what we speak as it was 500 years ago, something like that. Don Prothero will be speaking on the future of civilization based on energy sources and what kind of will we still be in the peak oil crisis in 500 years? It'll probably be gone by then. Greg Benford and David Brin, both science fiction writers and physicists, will be talking about colonizing the solar system and the far, far future of civilization. And then on in the midday Sunday, Richard Dawkins and I will be having one of these in-conversations, which Richard likes to do now, in which you explore ideas, and we'll be talking about the future of religion and things like that. You can tell he's thinking about religion right here. He's and then our big event coming up, our next big, huge Geo Tour a year from this summer, going to Great Britain. Donald Prothero will be telling us about that because he's leading that trip. Don Prothero.
I made sure to wear my Mammoth Fest t-shirt here in honor of our speaker. So anyway, not this coming summer, but a year from this coming summer, 2016, is our next big Geo Tour that we're planning to do. We decided to pull out all the stops and do something we hadn't ever done before, which is travel abroad, which is meant it's a little more complicated than usual. But we're also trying to do a trip that's not just unusual for us, but it's probably no one would also ever offer it. No one ever will offer it again. It's a one-of-a-kind trip. And it'll focus on the highlights of science and skepticism, and especially geology and paleontology of the United Kingdom. So it runs the last half of July of 2016, July 13th to 31st. We'll meet everybody in London at the hotel, which will be our starting point, and then we'll do a big loop through the British Isles and end up in Glasgow. I'm told that you can get your round-trip tickets between London and Glasgow in any part of the world just as cheaply as if you went round-trip to London, so we'll make it work that way. And it'll be a giant coach tour. We have now sold out the first coach as of like 3 days after it was advertised. It sold out the first coach immediately. We're nearly done selling out the second one. So if you're interested, you better move fast, because we will probably sell it out in a couple more weeks. Certainly after our May meeting, as we advertise it there, we'll probably do one more email blast, and that'll probably close the buses for the trip. The prices, it says up here, all lodging, all the 17 nights, I believe it is. All breakfasts are included, 9 lunches, and a bunch of the dinners are included. Transportation fees and admissions, the tour guides on both buses, and some of the dinners. I myself will be there for one of the buses, and then I've gotten Dr. Darren Naish, who's a paleontologist and geologist from Britain, to be our co-tour leader, who is actually better at this stuff than I am, since he's actually British. And he and I will switch back and forth between the buses, so there will always be a PhD paleontologist on the bus at all times. okay? No matter which bus you're on, you will not be shorted because we will switch back and forth, okay? So these will be sites that focus on history of skepticism, history of science, important history of natural history, basically, as well as some more conventional destinations, so it's not all learning all the time. So there'll be things, of course, like Stonehenge, which is legendary, of course, and we'll take a cruise on Loch Ness, so bring your binoculars and your camera, and we'll stop for a full day and a half in Edinburgh, the home of the Scottish Enlightenment, where geology was born. And we'll also see, for example, the man in the upper right there is David Hume, who could be considered probably the first great skeptical philosopher, who was a contemporary of those other great Scottish Enlightenment scientists and philosophers. As a geologist, we'll be focusing on interesting geology sites, and we'll do enough zigging and zagging across Britain. You'll cover nearly all the major areas there, all the way from the White Cliffs of Dover on the south end of the British Isles to the North Sea coast up there in the Highlands of Scotland, and lots of stuff in between. So there'll be lots of cool stuff to see, and I'll personally try and make sure we see some of the very important sites that led James Hutton to pretty much put us in the modern world of geology. And there are also things about the economic history of Britain connected with geology. So we'll visit a museum of a coal mining in Wales, where my ancestors once mined back in the 1830s. We'll be visiting a slate mine in Wales as well, which was once a big booming slate producing area. And we'll be visiting the famous Cornish tin mines in the tip of Cornwall as we're on our way out to Land's End, the westernmost point of United Kingdom. We'll also focus on paleontological sites of importance. So these on the left here are the famous 1851 models by Waterhouse Hawkins when they thought dinosaurs looked like big sprawling lizards. Those big structures are still on display in a park in the southeast suburbs of London. We'll visit those and some other important sites where paleontology was born, like the first dinosaur discovery site and so on. But also on the lower right, we'll visit Lyme Regis, the famous fossil coast of Dorset. And we've got at times, so we'll go in there in low tide, and the entire group will be walking along the low tide exposures just west of the quay there at Dorset, where if you ever watch French Lieutenant's Woman, that's where that's all set, by the way. And we'll be looking at the ammonites that paved the ground there at low tide. And then landmarks in the history of science and the philosophy of science. So we'll visit Charles Darwin's house and take a tour. We'll visit sites associated with Alfred Russel Wallace. The lower left there, we'll be actually visiting Isaac Newton's house, where Isaac Newton actually did most of his greatest work. For 2 years, he avoided Cambridge thanks to the Great Plague. He was actually doing his most creative work when he was sitting at home. And the apple tree, the legendary apple tree, of course, where he supposedly got his insights. So it's going to be a quite interesting mix of those kinds of things. We've also tried to get the nicest accommodations that we can put together for this kind of thing, including one overnight stay in Lumley Castle up in Durham in northern England. And we'll have an Elizabethan-style dinner apparently that night when we're at Lumley Castle. So that'll be quite cool to do. So it's going to be an interesting mix of a lot of different kinds of things, and I hope you'll all join us. I believe there's still a flyer, and I'm sure there's a flyer on the counter over there, but you can go to the skeptic.com site and pull down the GeoTour links, and it's the top link, the most recent one of all. And as I said, if you're interested in doing it, it will definitely be sold out by the month of July, probably sooner than that. Thank you.
Thank you, Don. So, some of you may have noticed last week that the Pulitzer Prizes were given out, and the winner in general nonfiction was Elizabeth Gilbert's book, The Sixth Extinction, which is blurbing this book. So, of course, one solution to the sixth extinction is stop driving species to extinction and save the ones we have, but of course, there's a lot that are already gone. Ergo, how to clone a mammoth. And I first heard about Beth Shapiro through my friend Stuart Brand, the creator of the Whole Earth Catalog, and now he's head of the Long Now Foundation. If you don't know the Long Now Foundation, it's terrific. After you support the Skeptic Society, I encourage you to check out the Long Now Foundation. They get people to think long term. They have this 10,000-year clock and so on. This idea that we should the expansion of rights that I talk a lot about, one of the areas is to expand the rights of future generations to have a place to live, that's reasonably healthy and whole, like our planet. So that's why I was intrigued by this idea, the science of de-extinction. Beth is a professor of ecology and evolutionary biology at UC Santa Cruz. She's written quite a few, of course, technical papers, but popular papers as well. And this is her first book, How to Clone a Mammoth. And she's the 2009 recipient of a MacArthur Genius Award. So with that, please help me welcome Dr. Beth Shapiro.
Are we good? Yeah, excellent. So thank you, Michael, for inviting me, and thanks to all of you for coming out here on a lovely Sunday afternoon when I'm sure there's many better things that you could be doing with your time. As a way of introducing myself, I thought I would start off with something that is kind of, I wear a lot of hats in my life and in my career, and one of them is that I'm a National Geographic explorer. So I've done some work kind of putting little pieces together that they use in educational materials, and done some kind of terrible documentaries as well. Well, documentary is kind of a nice way of referring to some of these. But anyway, this is a piece that we shot when we were up in the field, I think it was 3 years ago now. And it was shot by one of my students who was up there with me, which is why the audio is so terrible. So bear with me for a moment. But this is kind of what it's like to be me in the field trying to find mammoth bones. Vertebra.
The neat thing about this is that these are small pieces, which means that instead of wash down the stream, these pieces are actually still frozen in the permafrost. You can't get them out at all. So you can be really well preserved. Just heard that big splash of water back there. That means it's all broken through. Here comes the water. You better get out of here.
So, to be fair, that water is really gross, right? So what that is up there is, that was shot near Dawson City in the Yukon Territory in Canada. And those hoses that you saw were active gold mining community. This is called placer mining. So during the snow melt, all of that water collects into these big holding ponds, and then they pump it up using these big high pressure water hoses and wash away the dirt that has melted because of exposure to the sun. And then you get to the next layer of frozen dirt. They wait for a little while that melts and then wash that bit away. They're trying to get to the gold-bearing gravels that are beneath all of this frozen dirt. But while they're doing that, all of these bones come washing out. So we come up and kind of chat to the miners and look around for the bones and see if we can find them. And it's a really nice symbiotic relationship that we have with the mining community up there. But the dirt is frozen permafrost. This is dirt that plants and animals and everything else has been frozen and are now rotting in for probably anywhere between the last 20,000 to 700,000 years. So it's really gross, right? It smells disgusting. And the last thing you want is to be covered with this completely disgusting water and then out in the field for another 6 weeks. So yeah, there you go. So I'm a biologist. Sometimes I say an evolutionary biologist or a molecular paleontologist or just a paleontologist or genetics or something like that. I mean, I do lots and lots of different things, but I end up, mostly what I do is I go up into the high Arctic and I collect bones, and we extract DNA and other molecules that are preserved in those bones. And you might wonder why someone would do this with their career, so I'll start off by just kind of explaining a little bit of that to you. So we know that we're facing this period of rapid climate change right now, and when people think about climate change and how this is going to affect us and the species that we co-inhabit this planet with, we often see pictures like this, where we have changes in weather patterns causing severe droughts, or extreme storms with lots of damage, lots of species potentially on the brink of extinction. And of course, the media has a wonderful take on this, which is that we're all going to hell in a handbasket, right? It's clearly everything is just going to be a complete disaster from now on. So if you're a biologist and you want to study this kind of thing, a main question that you might be trying to ask is, how will species and populations and ecosystems respond to the climate change that is going on today and that is projected to occur over the next decades? So if you follow the climate change literature at all, one of the plots that you've very often seen, most likely, is this here. This is often called the hockey stick graph. And this is called a hockey stick graph because it kind of looks like a hockey stick. Here we have this thing here and the end of this right here. This line here is like some standard 1960s level climate, temperature. And then this is a proxy for what the temperature has been doing for the last 1,000 years. So it was declining a little bit with lots of noise around here. And then within the last couple hundred years, absolutely shot up. And of course, there are projections to where the climate is going to continue, temperature is going to continue to increase. This, of course, is not the first time in relatively recent geological history that the climate has increased really rapidly, right? In fact, if you extend the same kind of plot back now for the last 50,000 years instead of just the last 1,000 years, and again, this is just the standard everyday temperature, and it only goes to 1,000 years ago on this plot, you see that the climate was very cold for a long time. This was the Ice Age. Here's the peak of the last Ice Age. And then here we have this incredibly rapid increase in temperature, an incredibly rapid increase from 8 degrees below where it was in 1960 up to about present day temperature. We've been working a lot at this site in the Yukon, and what we see, in fact, is that this particularly rapid increase of about 4 degrees most likely happened over the course of several decades. That is a very fast increase in global temperatures. So what I do, and what other people who do the kind of work that I am working on do, is go back in time and ask the question, how did species and populations, communities and ecosystems respond to these past periods of rapid climate change, with the hope that we can use this information that we learn. So why do some species move or adapt? Why do some species survive and others go extinct? To help make more informed decisions about what we can do with the limited amount of resources we have to deal with climate change and species extinctions that are predicted for the present day. So I work in a field that's called ancient DNA. Pretty kind of clearly defined what it is. We get DNA out of old stuff, right? I work mostly in this place called Beringia. Beringia is this major landmass right here that spans about from Canada's Yukon Territory across here to the part of the world that Sarah Palin can see from her backyard, right? And you see that there's this lighter color right here, and the lighter color is because during ice ages, when lots of the water on this planet was taken up into forming glaciers that sat on top of the continents, the sea level was lower than it is today, and all of these lighter areas were land, right? These were land masses, and it formed a bridge that connected North America and Asia. And lots of different species used that bridge as a corridor to move back and forth between the two continents, including, of course, our own species that moved after the last ice age from Asia into North America. So today this area looks a bit like this, especially if you're far above the tree line. This is actually in north central Siberian Taimyr. I'm in that helicopter flying over top of this, where you have these large lakes and kind of scrub grassland with low-lying grass if you're above the tree line. But during the last ice age, it looked like this. This area was an incredibly rich and diverse grassland that was home to an equally rich and diverse community of plants and animals. Many of these iconic animals of the Ice Age, like mammoths and mastodons and giant sloths and hypercarnivorous bears that stood 14 feet tall, and a giant beaver, which stood 5 feet tall and is the funniest extinct animal. It just is, right? So we fly out into this place in, top-notch, absolutely top-notch transportation machines. You can see that some of our helicopter windows here are missing, which is fine, It's better for smoking when you're sitting on top of the gas canisters if the windows aren't there. We stay in 5-star accommodation when we go out into the field. I took this picture by stepping back from my tent and unfocusing the camera. Those are mosquitoes. And we wander along, and this is back in the Yukon in Canada where we shot that initial bit of footage that you saw. And here they are washing down this permafrost here, trying to get to the gold-bearing gravels beneath. Some of my folks are standing around here trying to look for these bones that are coming out. And we wander along here, and we pick up bones. So in a single day of walking along these places, we might get 7 or 8 bags that look like this, full of bones. We can get tens of thousands of bones in a summer coming out of this permafrost area. These are mostly mammoth and muskox and bison and horses and caribou, but we also find small numbers of carnivores like wolves and bears and lions and things like that. There are just many fewer carnivores, so their bones are harder to come by. We take a bit of DNA from those bones and take we take a bit of powder from those bones, take it back to the lab and extract DNA. And we've learned a lot of interesting things over the last couple of decades doing this work. We've learned, for example, that bison and horse and mammoth populations all peaked and declined at different time points during the last ice age, and were really keenly tied to the type of habitat that they need. Bison, for example, need rich grasslands in order to survive because they're ruminants, and horses don't. So maybe that can help to explain why bison persisted in the southern part of North America when horses animals went extinct. We watched bears and other populations of carnivores grow in size in response to growing populations of herbivores and disperse really long distances over incredibly short periods of time. And we're starting to get to the bottom of why some species, like caribou, survived. They didn't live anywhere near humans were. And other species, like cave lions, did not. And we publish these papers and we get a lot of attention because it's kind of cool. They're big animals, they're pretty awesome, and they make kind of nice headlines. And we get lots of telephone calls. And people, students and people from the public and the media call. And I'm always really excited to talk to them about my work, but they only ever have one question. To be honest, it's really depressing. So that brings us to de-extinction. So this is what people have decided to call this kind of new field or idea of bringing extinct animals or extinct traits back to life. Now, many of us first heard of de-extinction in the mid-1990s when we saw this brilliant movie, which should tell us, of course, that if we do bring extinct species back to life, there's no chance it could all go horribly wrong. And so I will take the next, 30 minutes or so and try to walk you through the process of how we would actually do this. How would we bring an extinct species back to life? And I'm going to focus on a mammoth, but I do talk a little bit about different species, and every species has different technical and ethical and ecological challenges associated with bringing them back. But I'm focusing on the mammoth just because most people seem really obsessed with it, probably because we can't clone dinosaurs. Sorry. We can't clone dinosaurs. There's no dinosaur DNA surviving in any dinosaur bones because they're made of rock, right? Once something is a rock, it doesn't have DNA in it, okay? So don't believe any of the crap that you read in journals like Science and Nature about dinosaur DNA, right? Anyway, how to clone a mammoth. So the first way that you might consider bringing a mammoth back to life is to clone it. I have clone here in inverted commas because clone is this specific scientific technique that people are thinking of. And this scientific technique is actually called somatic cell nuclear transfer, right? This is cloning, but in science words. Cloning is the process that's brought us in the mid-1990s Dolly the sheep. And I'm sorry this slide is a little bit washed out here. It looks better on my computer, but hopefully we can still kind of walk through this. So this is how cloning works. So here we had the mom of Dolly, or the clone of Dolly here. And what scientists did was they took some somatic cells. So we have two different types of cells in our bodies. Germline cells, which are sperm and eggs, and somatic cells, which are everything else. Sperm and eggs fertilize, create something that is capable of becoming an entire organism. Somatic cells are pretty clearly defined what they do, and the only thing they can do by the time they're a mammary cell in this particular case is be a mammary cell. They've turned off all the other genes that they don't need to be a mammary cell, and they're a mammary cell. So the trick to cloning is to convince that mammary cell to reset itself, to go back to some primordial state where it no longer has any instructions about what type of cell it is, and can therefore become every type of cell in a whole organism. So they did this by taking these cells and putting them in a dish in a lab and stressing them out, starving them of nutrients so they got totally stressed out. At the same time, they took egg cells from a different type of sheep. This egg cell was not fertilized but had the maternal genetic material in it, and they sucked out that nucleus. They sucked all the DNA out of that egg cell. So now you have an egg cell with nothing in it. And then they put these two cells next to each other, the stressed out mammary cell and this egg cell with nothing in it, zapped it with electricity, caused the cell membranes to burst, and this nuclear material from the somatic cell dumped into the egg. Then the egg proteins themselves have a kind of magic property where they can, after zapping it again, they can figure out how to, it will actually cause that nuclear material to reset. So by putting this stressed out DNA into an egg cell, they were able to convince that cell not to be a mammary gland, not to be a mammary tissue anymore, but instead to be able to create every cell in an organism. So this went on, and they implanted it in yet a different variety of sheep, and the pregnancy was successful, and Dolly, who is a genetic clone to the donor of the mammary cell, and neither to the egg nor to the mom, was born. Now, this worked, but Dolly was one in about 277 different tries that they tried to make it work. So it does work, but not very well and not very frequently. So How would we do this in the case of a mammoth? Well, we would go out and we would find a really well-preserved mammoth, and we would take a cell out of that particular mammoth, and we could then, stress it out, insert it into an egg. It does this little magical thing, and then we get this thing developing, put it in a mom, get it in the belly. We're going to do all this kind of stuff. It's going to go out into the, Right. So the first step, find a living mammoth cell. Well, it turns out that if a species has been extinct for around 3,500 years, there aren't any living cells. Right. So we find some incredibly well-preserved remains up in the Arctic. And some of you may have seen on the news this story about a really well-preserved mammoth mummy that was found, I think, in the New Siberian Islands that had a substance associated with it that they said looked like blood. Yeah, you've heard of that study? It wasn't blood. Yeah. Anyway, this is really well preserved, but still there are no living cells. And that's bad for cloning, right? Because if you don't have any living cells, you don't have any complete genomes, and you can't actually do this somatic cell nuclear transfer. So once an animal dies, what happens is, if it's a mummy in particular, these guys look incredibly well preserved, but remember their guts are full of bacteria, this microbiome that's really important to us. And as it's being preserved, the guts will burst, and all those bacteria will go through the bloodstream and get into the different parts of the cells. These are living bacteria that eat living stuff, right? These things will just catabolize, eat up all the DNA that's in there. So the enzymes within the body and then in soil bacteria that are buried in the soil will actually start breaking down the DNA sequences in every cell pretty much immediately. While we're alive, this happens, but we have proofreading mechanisms that are energy requiring, of course, that will go in and fix the damage from UV radiation that we get when we go outside, so we don't get cancer every time we walk outside. Well, once the organism is dead, these proofreading mechanisms don't work anymore, and the cellular structure and the DNA starts to break down. UV radiation, freeze-thaw, water, oxygen, enzymes from things in the soil, these will all act to break down the DNA so that even within a few hours after death, there will be no way to clone that particular species. So we cannot clone a mammoth. Thank you for coming. Just kidding. All right, so we all might have seen the news last week that the complete genome sequence of 2 mammoths was finally published and assembled. So we have all of these, we have this big long 4 billion letter string of ACs, Gs, and Ts that contains all of the information that we would need to make a mammoth. So why not just do that? Why not just sequence the DNA from scratch? And then after we have that sequence, we can get it into the chromosomes and get those chromosomes into the cell and then do that thing where it does the stuff and then you have the, So the problem is, this is the paper that, actually it isn't out yet, but the news picked it up last week. The problem is that even if we have as much of the sequences from this particular mammoth as we possibly can, we still don't have all of it. We still don't know everything that's in there. The complete genome that everybody's talking about is not actually a complete genome. And I know that's really terrible to say, but who knows how many complete genome sequences of any vertebrate organism, any eukaryote, are totally known? Any vertebrate? Let's go there. How many complete genome sequences are there? 0, that's right. Even humans, even our own species does not yet have a complete genome sequenced. Now, I'm being a little bit unfair. We know most of the human genome, and we know most of the part of the human genome that has the genes in it. But there are parts of the genome that are just these really tightly wound repetitive things that no existing sequencing technology can actually see through. So we don't know towards the center of the chromosomes where they come together, and towards the end of the chromosome where the telomeres are. We don't know those sequences. We don't know if they're important, right? There might not be anything useful in there, but we don't know because we haven't sequenced it. The situation is a little bit worse for a mammoth, and that is that not only do we not have these little bits and pieces, but we have to assemble the sequence in a funny way. So to explain that, first I'm going to go through 2 reasons why you can't really get a complete genome sequence from an extinct animal. First, the sequences that you actually get out of an ancient specimen are really short and really damaged. And that's because of all of those processes I just talked about, where UV radiation is hitting the DNA, and the freeze-thaw action of water, and oxygen, and the enzymes from other different things, from the soil microbes and the gut bursting, are all going to break those DNA strands down into smaller and smaller fragments. So if you think of chromosomes as these big, long strands of party streamers, in fact, what we get when we extract DNA from a mammoth bone is something that looks more like confetti, right? But maybe not even confetti that's this nice, you know? Maybe it's confetti like the day after the parade, when it rained the night before. And there were mammoths in the parade, and passenger pigeons flying overhead. Yeah, that kind of horrible, gross confetti that you wouldn't want to clean up. The second reason that it's hard to sequence and assemble a complete genome from an extinct species is that the samples that we extract don't only have mammoth DNA in them. They have all sorts of other types of DNA in them as well. So if you imagine that I've taken a piece of my own hair or maybe a cheek swab, and I extract DNA and then sequence everything that was there, most of what I get out of that will be my own DNA, because I'm alive, my DNA is in good shape, I'm not too contaminated by other stuff, hopefully. When you extract DNA from a bone that's been sitting in the soil, and this is from one of the first mammoth bones that we actually sequenced, everything that we extracted from, we found that about half of the sequences we recovered were mammoth DNA, and the other half were things like unidentified soil microbes, or actual soil microbes that we already knew what they were, contaminating sequences from humans or other laboratory things that you might find in there. There's a lot of crap in these samples, right? So instead of getting something that looks really nice like this, or even the day after the parade, in fact, what we get is like, you know, the day after New Year's. It's really awful. It's really terrible. And we have then this challenge of trying to sort through all of that stuff and figuring out what of those sequences is mammoth and what of those sequences is all kinds of stuff that we want to throw away. The way that we do that is we already have the genome sequences for living close relatives of many of these extinct species. We know the genome sequence of the Asian elephant, which is the closest living relative of a mammoth. And if we were working on the Neanderthal genome, now the Neanderthal genome was even harder. The first Neanderthal DNA sequences that were published, the first shotgun, well, they call them shotgun libraries because you sequence everything that's in the library. The bones that they used to generate those data were less than 1% Neanderthal. So 99% of what they sequenced was crap they needed to throw away, right? So this is hard. This is a hard thing to do. But we use the genome sequences of these extinct things, of living things, as a scaffold onto which we can map these kind of broken up little tiny fragments of DNA. That's what's there. Now this is a problem, right? Because if there are parts of the genome that are very different between an Asian elephant and a mammoth, we won't get them at all using this because we won't have the map to use to match those sequences. Instead, we'll just throw those sequences away thinking that it's just other stuff that we sequenced from the ground. And one might think if you really wanted to know what's different between an Asian elephant and a mammoth, those are the sequences that might be most important to understand. So just because we have a complete genome mapped to an elephant genome, which is what the published data are, does not mean we have the complete genome sequence. The other problem with this particular approach is that a DNA sequence is not the same thing as a living cell. Your DNA is wound up into these chromosomes and they got wrapped around histones. It's really tightly wound. The chromosomes are inside the nucleus. We don't know how to do any of this. Even if we could synthesize strands of DNA that were as long as chromosomes, which we can't do yet with very high fidelity, we couldn't turn those into chromosomes, and we couldn't get those chromosomes into a cell. So it looks like this particular plan B of sequencing the genome is not going to work. Of course, there is another way. So plan C, engineer a mammoth. This is the way that this particular type of de-extinction research is going to work. So what I mean by engineering a mammoth is really very similar to cutting and pasting your way from an elephant to a mammoth. So we have the DNA sequence of the Asian elephant, and we have this mapped sequence of a mammoth. And using that information, we can figure out where these two animals are different from each other along their DNA sequences. So imagine that you could design a tiny little machine that could go into those genomes inside a cell, a mammoth cell, get into that genome and find a particular bit of that genome sequence. And imagine you could also give that little machine another bit of synthesized DNA, small enough to where we can do it, right? Unlike sequencing and synthesizing a whole chromosome. And you could inject both of these things together. And what that machine is able to do then is to find in that DNA, and then you could take these little things and inject them, right? See, this is kind of trying to get this obvious in here. And it would move around that little cell and find the bit of the DNA that you designed that machine to find, and then chop out the elephant version of the DNA sequence, and then use that piece of DNA it brought with it to insert in its place the mammoth version of that particular cell. We can do that. We have those machines. It's called CRISPR-Cas. This is a type of genetic engineering or genetic editing modifying kind of machinery that you've heard a lot about recently. Last week or the week before, there were some Chinese researchers who used this technology to edit human embryos, and it caused a big stink, right? This is a scary, powerful bit of technology. But one of the things that it might be able to do is edit the genome of an elephant in a cell, in a dish, to where it has swapped out some of the places in the genome, the elephant version of a gene for the mammoth version. So you stick this thing, this is your little machine, this whole enzyme here, and this is the arm that recognizes specifically the place in the genome you want to change. Let's say we want to swap out DNA right here. And with it, we stick in our DNA sequence, which is just a synthesized bit of mammoth DNA. This thing finds that bit of DNA and it cuts it. It acts like a molecular scissors and just cuts it in half. And then it uses the cell's own repair mechanism. Cells don't like when they have broken DNA. So there's all this evolutionary mechanism that exists to fix breaks in DNA. So you trick the cell into using its own inherent evolved processes of fixing itself to stick in the place of this elephant version, the mammoth version of DNA sequence that you've stuck there. And you end up with an Asian elephant, right? An elephant cell in a dish that's a little bit mammoth-like, right? This is how this works, pretty straightforward. So what do we change, right? If we're going to go in there and we're going to take an Asian elephant cell and we're going to change bits of its DNA, swap out places for mammoth versions of genes, what do we change? We know there's been about 6 million years of evolution separating the Asian elephant and the mammoth. Right? And over 6 million years of evolution, a lot of differences have accumulated. So the places where people have started looking are in the gene sequences, not outside of the genes, but in the gene sequences for places where many mammoths are all the same, many elephants are all the same, and mammoths and elephants are different, right? So these are clearly places where there's something that has happened along that evolutionary lineage to mammoths that is mammoth specific that we might want to change. Something, for example, might be there was a gene that was discovered, or a series of differences that was discovered a few years ago by Kevin Campbell and his lab in Manitoba. And they were looking at red blood cells, the hemoglobin molecule in red blood cells. And they found that there are 3 differences that change the sequence of the protein that are different between woolly mammoth version of the hemoglobin and the Asian elephant version of hemoglobin. And importantly, they measured, once you make these changes in a cell, in a dish, they measured what those changes did, and it turned out they made the hemoglobin, the red blood cells, better at carrying oxygen around the body at lower temperatures, right? So this is something that is pretty cool. It's obviously something that if we wanted to take a tropically adapted Asian elephant and stick him somewhere cold, this would be something we want to change. So what else? So there are a lot of different things that we might want to change if we're thinking about turning an elephant into a mammoth. And there actually is a group led by George Church at Harvard, the Wyss Institute, and they have been doing this. They've actually changed so far 14 different genes, most of them associated with temperature regulation. He's also looking at things like how you can get the hair to grow, or different dietary preferences, or immune-related genes, all these kinds of things that you might want to change in order to take an elephant and turn it into a mammoth. So far they've done 14 genes. They've created in a dish, in a cell, in a lab, something that is about 0.001% mammoth. And remember, this is not an actual mammoth. This is cells growing in a dish in a lab. That is very cool. But what it means is that we can do this. We can get the genome sequence, which we already have. We can use this crazy genome editing technology, and then we can do all this other stuff. We have the genome sequence, right? That's been published very recently. We have this CRISPR-Cas technology, and we already have somatic cell nuclear transfer, which is something we can do. That will bring us to phase 2. Phase 2 is where we have to then find a mom, grow the baby up, have it be born, and find a place for it to live. The challenge here, I think when a lot of people, especially when we hear a lot of the hyperbole that we hear in the news, is that people kind of get fixated on phase one, and they're like, whoa, we can do this kind of crazy stuff. We're definitely going to be able to do it. When they forget that this second part here is going to be maybe even harder than the first part of getting these things. First of all, you have to find an appropriate surrogate mom for something that's extinct, right? So is it going to be, what kind of challenges are we going to encounter when we're trying to grow one species up inside the developmental environment of another species? And what if that species is really distantly related? So there are some extinct species, like a moa in New Zealand, whose closest living relative is something like 30 million years diverged. How many differences have evolved, and is it going to be too many to actually be able to use egg material from a living species that's that different from the species you're trying to bring back? In some cases, there might actually be physical constraints that would cause a problem at this particular stage. I say that a picture is not really required or desired, but I do have a picture. Don't worry, it's not gross. It's just a picture of Steller's sea cow here. This is a Steller's sea cow. It's an extinct animal that lived off the coast of California, all the way up across the Aleutian Islands into the Commanders. This was an enormous thing. Its closest living relative is the dugong, right here. This is a dugong, right? Based on the ratio of size at birth to mom, if the dugong were going to carry a baby Steller's sea cow to term, at birth it would be bigger than its mom, right? That's not going to work. No. There are also going to be environmental differences. So we know very well that we as people are more than the sum of the A's, C's, G's, and T's that make up our DNA sequences. We are a product of our DNA sequences and the environment in which we live. If we were only a product of our DNA sequences, identical twins would be completely identical all the way throughout their life. And yet we know that when identical twins are born, they're very identical, and they start to diverge throughout their lifetime because they're exposed to different environmental cues, different diets, different exercise regimes, different stress, right? What does growing up inside the belly of an elephant do? All of the exposure will be to elephant hormones regulating gene expression, to the elephant mom's diet. When it's born, it will be fed an elephant diet. You know, elephants, when they're born, they consume some of the poo of their moms so that it resets or sets up the microbiome community in their guts, right? We don't know what that community looked like in a mammoth, and we don't know the effect of having the wrong or the elephant version of that might be. It will live in an elephant society, presumably, and eat an elephant diet, or maybe whatever we can actually figure out that it might want to eat. You know, maybe they like to eat Christmas trees, who knows? And of course, then, there will be other considerations. We know, for example, that elephants fare very poorly in captivity. They suffer a bunch of different physical and psychological problems when they're in captivity. They often fail to reproduce. If they do, sometimes they injure or even kill their offspring. Until we've figured out how to take care of, how to meet the emotional and physical needs of elephants in captivity, I think mammoth de-extinction is a no-go. This is in my mind. The ethical considerations of this are far too great, the rights of these elephants. So there will be ethical considerations in all of these different de-extinction projects, and they're going to differ depending on what species they are. But there definitely will be ethical considerations. And then finally, there are ecological implications of recreating something that's not here anymore, right? When a species goes extinct, the environment, the ecosystem in which it lived does not remain the same with this massive gaping hole just waiting for us to reintroduce that thing into. The ecosystem adapts to the absence of something. Other species move in, the niches change, the whole thing changes. We can do sophisticated risk assessments to try to figure out what the effect of introducing a mammoth into an environment where they aren't might be, but we're never going to be able to completely know what all the risks are. I'm not arguing that this is a reason against it. I think there are many reasons to take risks such as this in conservation, but there are ecological implications. For some species, there will be absolutely nowhere to put them. Imagine bringing back passenger pigeons. This is a species that flocked in 1 billion individuals across the eastern coast of North America in kind of through the Midwest and up through the Maritimes in Canada. Where would we put 1 billion passenger pigeons? Where would they go? Who would really want to see this flock of birds coming at them that they knew was going to darken the night sky for days as they passed overhead? Not somebody who just washed their car or their house, probably. So de-extinction, can we do it? Well, the answer actually right now is no, we can't do it. We can't bring any species that's extinct back to life yet, right? Technology is developing, and it's developing at a really, really fast pace. And probably someday there will be a time when we can bring extinct traits, at least, back to life, even if we can't recreate something that is 100% identical to a species that used to live. But a big question that I think we probably should be asking ourselves is, should we bring extinct species back to life? I've thought a lot about this, obviously, and people keep asking me to have an opinion about this, and my opinion is very, very certain. I have no idea, right? And there are a bunch of different de-extinction projects that are underway, and they all have different ethical and moral and social implications and challenges that they're facing. And some of them, maybe there are compelling reasons to bring them back, and others maybe there aren't. But I do think that the fundamental question that we should be asking before we do anything with any of the species, yeah, there's a stellar sea cow, just to give you the size of perspective there of how big those things were, right? Anyway, the question we should first ask ourselves is, why? Why do we want to bring an extinct species back to life? So just for these last few minutes, I'm going to return to the mammoth, okay? And let's imagine that the technical considerations, we've solved them all. The ethical things, we're not going to think about for a moment. I've already said that ethically, I don't think mammoth de-extinction should happen. But what really are the compelling ecological reasons for bringing a mammoth back to life? Well, I have two. So first of all, there's this place up in northeastern Siberia, and it's right about here, outside of Chersky, that Sergei Zimov, who's a Russian Academy of Sciences scientist, has been establishing. He started buying up land in the mid-1990s, and he's growing the space of his Pleistocene Park, and he's preparing Pleistocene Park for the megafauna that lived there during the Ice Age. And so far he has horses, bison from Canada, 5 different species of deer. Every now and then he lets a bear visit to see if they can handle it around. And he's noticed some really interesting things about what happens to this tundra landscape with these grazing herbivores present. So this is a picture that Sergei took. He has these exclosures or enclosures, depending on which side you're on, where he has grazing herbivores on one side and doesn't have them on the other. And this is a picture that he took in early spring, just after the snow finished melting away. And you see there are big differences between this part of Pleistocene Park, where there are grazing herbivores, and this part where there aren't. Here you have pretty much one species of grass that dominates. There's not very much biodiversity. There's not very much rich sources of food for the animals that live there. Over here, there's lots of diversity. You can't really see it, but you probably can see some of these kind of green bits of grass that are poking up here. So it turns out that just their presence, just the existence of these herbivores on this landscape has been enough to kind of spread around nutrients, recycling them, as they wander around, digging up and aerating the soil, move seeds around, and really create this rich grassland that's much more reminiscent of the type of steppe tundra, this rich grassland that was there during the ice ages, than what they have today. So just the presence of these herbivores changes this ecosystem into something that supports themselves, right? They create their own habitat. He's also seen species, individual saiga antelope coming to visit this particular location. Saiga antelope are an endangered species because there isn't very much for them to eat in Siberia. He's creating habitat to protect species that are in danger of extinction today. And I think that's pretty cool. The other thing that he's noticed, and this is now venturing in the realm of crazy, so bear with me. The other thing that he's noticed, not as if I've been crazy enough already cloning a mammoth, right? Anyway, I'm really going crazy now. He has noticed that the soil temperature underneath the soil here, where he has the grazing herbivores, is between 10 and 15 degrees Celsius colder during the winter than the soil temperature over here. Now, why might that be? It turns out that snow is a very efficient insulator. And the subsurface soil temperature of the permafrost is an average of the air temperature during the year. So the soil is exposed during the summer. It's relatively warm during the summer, somewhere like 60, 70 degrees, even up there during the summer. And the surface gets warm and it starts to thaw. And then the snow falls and it traps that heat inside that soil. So the permafrost is actually melting. The animals that are there come along and they dig up these rich grassy areas, exposing the surface to the very cold, 40 below at this point, very cold winter air. And therefore, the average temperature that is this soil is a lot lower than the average temperature without them, exposing the surface of the soil to the cold winter air. So Sergey argues that we should bring back these large herbivores because by exposing this soil and dropping the temperature, we're slowing the rate of permafrost thaw. Now, scientists have estimated that there is currently as much greenhouse gas carbon trapped in the Siberian permafrost as there is in the atmosphere. He says, Sergey says, that by slowing down this thaw, he could be slowing the rate of global warming. Right? Nuts. Told you I was going to nuts. But how cool would it be if it actually were true? So the last reason that I think, and potentially to me, this is the most compelling reason that we might think about this technology, and this is the reason that I really believe that we are heading somewhere positive by developing this technology. Many people, I guess few of us will be willing to accept a future without elephants, right? But elephants are endangered, and their habitat is continuing to disappear. We're having trouble protecting them in the places where they live because of expanding human populations and uncontrolled poaching. Elephants are in danger of going extinct. What if we could use this technology not to create a mammoth, because we can't, we're never going to be able to create something that's 100% identical to a mammoth, but instead to create an elephant that was able to live in a colder place, like where the mammoths used to live. We could then have elephants released into wild habitats that we could better protect, or didn't need to protect, like this Siberian permafrost, potentially using this technology not to bring mammoths back to life, not to save a species that's already gone, but to save elephants. And this, in my mind, is the most compelling use of this technology, not as a tool to recreate something that's gone. If they're gone, but as a tool that we can use to use evolution that's already happened, traits that have evolved in these extinct species, as a tool for preserving biodiversity, for conserving and protecting ecosystems and species and populations that are alive today, but in danger of going extinct. And that, I think, is the positive in de-extinction.
Very ambitious. So after your questions, you can get your books here and you can get them signed here. I should note that Princeton University Press really does produce beautiful books. There's a nice section of color plates in here. It's quality paper. Those of you who still care about real books, I do, and manage them. There's an extra cool thing. If you buy 2 of these, like this, you see, so they make a nice gift. And then we're actually also going up to dinner afterwards. This is sort of our tradition. Burger Continental is now out of business. They retired. And we're now going to El Portal Restaurant. It's a Mexican restaurant on Green and El Molino, just a few blocks from here. Green and El Molino. We'll be over there by about 4 o'clock. Or so our speaker's coming. We just hang out and have good Mexican food and margaritas. So that's always fun. So, okay, let's start off right here.
Interesting talk.
Thanks.
This question relates to parts B and C and phase 2. Assuming that you didn't have a complete DNA sequence of, say, a man.
Right.
From what I understand from stem cell research, is that there are these things called growth factors / marker proteins that are the things that turn on and off the genes within the cell. Now, we know that some of them come from the mother's womb, right? And some of them come from maybe encoded in the DNA, and we don't figure that out right now. So the question is, if you took the DNA and put it in, let's say, an elephant, you kind of think that, well, there may be the right marker protein, growth factors, whatever, that, a baby mammoth would come out. But if you put it in, let's say, a chihuahua, would the chihuahua burst because a baby mammoth would still want to come out? Or would the chihuahua's growth factors turn on different things that would make it come out like something different? And that leads to the second part of the question.
Wait. That's I'll let you get to the second part of the question, but that was crazy. I've said that was even crazier than stuff that I've said. I don't want to put an elephant inside a chihuahua because I can't imagine filling out the IACUC paperwork for that experiment, you know? Just kidding, there are other reasons not to do it as well. But, but I don't know the answer. And also we don't know the answer of whether or not these epigenetic tags that turn genes on and off, just with this difference between elephant and mammoth, are going to be so different that you're just going to end up with a mammoth, an elephant instead of a mammoth. We don't know. We have absolutely no idea.
And that relates to the second part of my question. Maybe there's researchers here. It seems like it sets up a set of Mendelian experiments where we should be taking little species that we have, like rats and mice and chihuahua, whatever, and taking their fertilized eggs and putting them in each other's wombs to see how each one affects.
Yeah, so there have been, I totally agree that we should be working on something that doesn't take 14 years to go from fertilized egg to sexually reproductive adult, because that's just too long for anybody's career to be doing this kind of work, right? And mice probably would be a good place to start, kind of thinking about how this reproductive differences might actually change the gene expression patterns and then what the phenotype, the physical appearance of the animal was. There have been successful births of one species that have been carried inside another species. So there's been a gor, which is a type of cattle, and a bantang that have both been born to a regular old domestic cow in Ohio. And the bantang, I believe, is still alive in the zoo in San Diego, actually, living just fine and looking like a bantang and not like a cow. But that's probably a couple million years of evolutionary distance, but we know that it works, right? The Bucardo project, the Bucardo sequencing project, this is a recently extinct type of ibex from the Pyrenees, right? It went extinct in the early 2000s. Before it went extinct, scientists who were working on it, conservation biologists, actually sampled a bit of cells, living cells, and managed to freeze them. So now they can do somatic cell nuclear transfer with this thing because they have living cells, not like a species that's been extinct for a long time. But they had real trouble getting fertilized eggs made from these cells, not fertilized eggs, but these kind of clone reset eggs from these cells to work successfully with ibex, another very closely related species. And this wasn't a genetic problem, this was a behavioral problem. So they couldn't actually get the closely related ibex to come down from the high ledges inside the lab so they could do this kind of stimulation to get eggs from them. They had to actually create a hybrid domestic goat ibex to carry that as the surrogate mom to be able to do that. They also couldn't do it with a regular domestic goat. That turned out, they think, to be just too much evolutionary distance. The problem there, though, is that this cloning process, somatic cell nuclear transfer, is not particularly efficient. So if you try 200 times and it fails, it might be because there's some barrier to it actually working, or it might just be because it didn't work that time. Right? So this is a very tough experiment to do with long-lived animals that require considerable care. I'm not sure it's fair to do it either to these guys. I mean, it depends really why, right? Again, coming back to why. What is the motivation for wanting to do this? Yeah. Anyway, move on. I should move on. Yes, here.
So this is probably a stupid question, but I think your answer would be good.
No questions are stupid.
And that's that you said flat out no dinosaurs, but the premise of Jurassic Park was not from the bones or anything, but from a mosquito trapped in amber, the blood that got it. So not necessarily dinosaurs, but is that plausible at all in any way to get DNA and genetic material from blood from a mosquito trapped in amber of any species?
No.
And then use it.
So this was so Jurassic Park, actually, when Michael Crichton wrote Jurassic Park, he acknowledged in the back of the book a group at Berkeley in Alan Wilson's lab called the Extinct Species Working Group. These were the very first people who were working on ancient DNA. So his ideas for Jurassic Park were motivated by this field of science that was in its very early days then, but that I'm now involved with. And one of the first things that people did in ancient DNA was try to get DNA from dinosaur bones. And they said, look, we got DNA from dinosaur bones. And they published it in Science and Nature. And it turned out to be human contamination, right? And then they did it again, and it turned out to be a contamination from a chicken sandwich someone was eating in the lab. And then it was amber. Then it was, oh, we can totally get DNA from amber. And of course, there was a series of papers published about DNA from amber. None of this stuff has ever been able to be shown to be true. In fact, about a decade ago, a team in London went to back into their collections, and they have lots of insects trapped in amber. I've actually done this as well, and one of the chapters in my book starts with my story of extracting DNA from amber, right? So So what this team in London did was they got amber that was really old, and also copal, and this is the precursor to amber. So the tree sap hardens, and first it forms copal, and then it forms amber over a long time. They got copal that was only a couple decades old. We know we can get tons of DNA out of stuff that's decades old. And they extracted DNA and amplified up kind of insect DNA. And they did get insect DNA from some of their amber and some of their copal pieces. Unfortunately, there was no correlation between whether those pieces had insects in them and whether they got insect DNA out of them, right? And the only explanation for that is that anything they were picking up was just contamination from somewhere in the lab. Amber is actually a very porous substance, and bacteria can get in it while it's soft. And the idea is that there is no way that DNA can survive in it for very long. It's also very hot, where you have this stuff forming. It's a terrible environment for DNA preservation. We work in the Arctic because it's cold there, and the bones are better preserved where it's frozen. So unfortunately, very upsettingly, amber is a terrible place for DNA survival. Turns out La Brea tar pits are also a terrible place for DNA survival, which is terrible because there's such an awesome collection at La Brea that it would be so cool to be able to get DNA out of. And they look so wonderfully preserved, but we just can't get the tar out of them, and the tar seems to really have destroyed anything that's in there, or at least inhibited our efforts to get DNA out of them. So sadly, we're stuck with bones and blood. I'm going to go back and then toward the front, so in the blue shirt.
What about mitochondrial DNA? Doesn't that sort of confuse the whole thing with all this? You're not getting mitochondrial DNA from whatever you're trying to be distinct.
Yeah, we're not. Mitochondrial DNA is something that we can sequence, and I said to George Church, this is the guy who's leading this de-extinction effort at Harvard, what if there's some incompatibility between the mitochondria and the nuclear genome that you're replacing? And he said, just change the mitochondria as well. Use the same genome editing tool to swap out the mitochondria. So, yeah, maybe that's the solution. In the pink, and then you.
We just saw in the last couple of weeks news stories about regarding the last black rhino male in existence. Is anybody actually planning to hopefully get DNA samples from these organisms and maybe do something to preserve them and build upon these techniques to allow us to bring back
Yes.
those animals? Good.
Yes, there is a whole institute in San Diego called the Frozen Zoo that's associated with the zoo. And for the last couple of decades, Oliver Ryder has been working to collect DNA samples from all sorts of endangered plants and animals and other things, and things that aren't endangered, and store them in this particular kind of really weird place. You walk into all these massive liquid nitrogen containers full of tissue culture, and they actually create growing tissue culture from cells from these organisms and then preserve them, so that once this technology becomes able to do something about it, we can do that. He's actually been working very closely with the rhinos and has all DNA samples from all of the rhinos that have been alive over the last 2 decades. So not only does he have rhinos, but he has a diversity of them as well. So when this technology were to come back, then he would be ready to go. And he's been planning on this for decades, right? He's a very forward-thinking guy. This Hawaiian shirt, yes.
You kind of answered my original question, but which was, how are we going to save the elephants long enough for the technology to keep up at the rate we're going? But what about using the technology to save our own species?
I don't think we're in any danger of extinction.
We don't know how fast the climate change, are we going to be able to adapt to the rapid increase in climate change?
We don't have to adapt. We can build things that'll stop. I think we're a special case here, and I think we should be thinking about slowing down rather than manipulating our own genomes to the point where we can do even more. I don't know. The planet will be fine no matter what. I think, if people who care about extinction probably really, a lot of people who care about extinction only really care about it in as much as it affects them personally. So it's their own extinction that they're worried about when they're thinking about any sort of biodiversity conservation. Probably not true of people in this room, at least I hope so. Sorry, in the white shirt.
In the Italian Alps, when they discovered the hiker that was hundreds of thousands of years old, number one, went through your mind, but number two, there have been genetic changes in the human species over thousands of years. And I'm thinking of cancer research today as an example of how to, the new immunology advances, that a lot can be learned from humans 3,000, I should say 5,000.
Yeah, he was 5,000 years
What
old.
went through your mind when you heard that, and have you fantasized about that?
I have a piece of him in my freezer. Which is kind of scary because,
Vodka in my freezer, but
yeah, so this technology, this CRISPR genome editing technology, is not being developed to save species. It's being developed to cure human disease, right? There's no money or energy going into developing this technology for the purposes that I'm talking about here. All of it is going into exactly as you say, trying to learn something about how human genome has evolved and maybe fix genetic changes that we know are associated with certain diseases. That's where the resources that are engineering or pushing this technology forward are coming from. And yeah, we can learn a lot. I mean, my whole research career is based on the fact that you can learn about how populations and species adapt to changes in climate or different community compositions over time by looking at their DNA sequences. So absolutely, I agree with you that we can learn a lot from people that lived a long time ago and from Neanderthals, you know? How much have we learned by looking at the Neanderthal genome and figuring out where exactly we're the same as Neanderthals and then where we're different? I mean, this is really, with the Neanderthal genome, we're starting to really be able to hone in on those evolutionary changes that make us different, that make us unique. And I think this is super exciting and fascinating research. Absolutely. There's more? Green.
I was going to pronounce, but I might just be wrong and really confused, but I was under the impression that Dolly was like born old, like there's something going on with the telomeres that made it where the sheep sort of didn't live very long after.
Yeah, there's been a lot of speculation about what happened with Dolly. I think, I'm not totally sure, but I think the latest of what happened with Dolly is that she died of a regular kind of virus that infects sheep in captivity. Yes?
So you mentioned from sequencing the La Brea bone because of the tar contamination. Let's say about 10 years ago now, they claimed they sequenced smilodon DNA in Siberian cat.
Steve O'Brien's group.
A lot thought to be contaminated.
It was a contaminant, yes. Yeah, that was Steve O'Brien's group at NCI, the NIH National Cancer Institute. Steve has always been obsessed with cats, which is probably why he now works in St. Petersburg instead of the Cancer Institute. But yeah, that was a really cool project, but it turns out to have been a contaminant. There's never been anything that's successful and been replicated come out of La Brea, which is very disappointing, and we will keep trying as the technologies get better and improve.
Okay, two more.
Right? Anyone else? Oh, here. And then down here.
Different part of your lecture. How much does your aquaflexion stuff in the permafrost, how much does that process degrade the specimens you're collecting? And would you rather have the ability to collect them without that hydrocannon thing going on?
That is actually, I mean, the way that we find these bones, the places that we find them are from these placer mining stuff. Or along the riverbeds, because as the snow thaws, the rivers in Alaska, these meandering rivers, will actually rise up and strip the same process, strip the sides of the permafrost off and deposit bones along the next corridor. I would rather find them from placer mining because often we find them still frozen in situ and we can see how old they are from where they are in relationship to different volcanic eruptions that have happened, that we know when they happened. And the other process of mining is incredibly destructive, where they go in with bulldozers and then you don't find anything, right? So that process is actually really good. We're finding bones that we can actually keep frozen. We managed to sequence and put together a complete genome sequence from a horse, just a regular old horse, that was 700,000 years old, right? And that was because we found it frozen at one of these gold mining sites. And we were able to recover that bone and keep it frozen. It had been in that dirt freezer for 700,000 years, and that's why DNA survived in it. If, you know, there's no way that if it hadn't been frozen for that time.
Russia found in Siberia frozen mammoth.
They found a lot of mummies, but the mummies, money, kind of counterintuitive, but mummies tend to be much more poorly preserved than isolated bones. And I think it's because when they're actually warm for quite some time and even get hot as the bacteria kind of explode and go all over the place, and that degrades the DNA. Whereas if an animal is stripped of flesh and the bones are quickly frozen in the permafrost, the DNA in those bones tends to be much better preserved. The key to this 700,000-year-old specimen is that 700,000 years old is the oldest permafrost that's known. There's no permafrost that's that old in Russia, right? And the DNA was in terrible condition. The average fragments were 40 base pairs long, the average fragment size, whereas if I extracted DNA from me, I could get fragments that are millions of bases long. So 40 is kind of crap, but it was there, which is what was most important. And this question here. Nope. Here. This is the last one.
It's just that are there different species of mammoths that have been found across
Yeah.
Siberian? Are there any that are seen as more suitable for those sort of
Yeah, so
de-extinction?
the mammoth species are really chronospecies. So paleontologists have named different mammoth species depending on how old they are. And kind of the dentition, you can see how the teeth change through time. The only one that we can really think about from this perspective is the most recent one, this Mammothus primigenius, the kind of late Pleistocene mammoth. Everything else is way too old, millions of years old. There would never be DNA in them. So there are different species, but it's kind of like the same animal evolving over time and paleontologists naming them different species as they're wont to do. So.
Let me ask you one last question. A completely unrelated topic to this, but something you mentioned worried me. All those papers published in Science and Nature, this one, throughout the 90s and the 2000s, none of them are true. What else am I reading in Science and Nature that in 20 years the speaker here is going to go, that stuff, it's all, is this a concern?
The arsenic life stuff, that was shown to be bad immediately after it was published. I was at a dinner. My partner, Ed Green, was the first author on the Neanderthal genome sequencing paper many years ago when it came out, and that paper won best paper of the year in science. So we went to this dinner at the AAAS meeting with the editorial board from Science, and the chief editor at the time asked, he said, so what can we do to make our journal kind of better for you? What can we do to you, the reader, the user of Science magazine, to make it better? And there was complete silence around the table. And then my partner, who sometimes doesn't have the right block between thoughts that come into his brain and things that come out of his mouth, said, what the heck was going on with that arsenic paper? And there was kind of dead silence. This is a paper that came out saying that somebody had discovered a new form of life that was based on arsenic, right? And it turned out that as soon as it came out, all sorts of researchers were like, whoa, absolutely not. Let me show you why this is wrong. This is ridiculous. How did it get published? And they defended it by saying, if they really are going to be on the cutting edge of stuff, they have to be willing to take risks. And if you only ever publish stuff that is always right, then you're not taking risks, right? And I kind of get that.
Okay. So 50%.
They have a pretty good hit rate, I'd say. Just these dinosaur DNA things work.
Okay, very good.
Thank you.